Hi, and welcome to this presentation. I'm Shane Herbert, and I'm the Program Manager for eDNA for Global Environment Studies, or EDGES. This presentation will be an overview of what eDNA is and some background on its uses. Then we'll go into the program that formed around it and an introduction to its research projects by the lead researchers themselves. So here is the EDGES team. The lead academic heads of the projects are Professor Ewan Harvey, who specialises in marine science, and Professor Morten Allentoft, who is the head of the Trace and Environmental DNA Lab. Moving to the right is Dr. Mattia Sacco and Dr. Nicole White, who also lead projects in the EDGES program, and from whom you'll hear from shortly. Moving to the bottom row, we have Shirley Luckhurst and myself, who also reside at Curtin University, followed by two key external collaborators in the projects, being Dr. Tiffany Simpson and Dr. Justin McDonald. So to get things started, I'll hand over to Professor Morten Allentoft to introduce eDNA.
Thanks very much, Shane. I will give a brief introduction to the topic of environmental DNA before we then move on to discussing the 5 different sub-projects in this program. So environmental DNA is essentially it's DNA found in the environment. And you can say DNA exists as molecules inside the cells of all living organisms, but when an animal or an organism is passing through the environment, some of its DNA will also end up in the environment. And this can be because, for example, some cells are being shed from the skin, or it can be from saliva or feces or urine. In many different ways you can think of where these cells actually end up in the environment, which means that the DNA is also being released into the environment, and that's when we call it environmental DNA. And this can be from large species, which you can easily observe, but it can also be from small microscopic or elusive species that are difficult to identify by traditional means in the nature. This can then be captured by filtering water samples or sediment samples, and you can bring those samples back to the laboratory, and you can extract the DNA that are present in these environmental samples. And then the trick is that you can actually identify which species are represented in these DNA sequences, because all species have unique DNA profiles. This is what has allowed them to evolve to different environments over evolutionary timescales, and we can utilize that information to actually identify which species are present in the environment because they have unique DNA profiles. That's the basic foundation for how to do environmental DNA studies. Based on this principle, eDNA, environmental DNA, can then become a biomonitoring tool because you can sample the environment, different substrates, whether this is sediments, whether this is freshwater, whether this is a marine environment. You sample the environment, you extract the DNA, and you figure out which species are present at that particular point in time. And this can, of course, be applied in many different contexts, as summarized on this slide. I'm not going to go through all these examples here, but just it becomes a tool, for example, for looking at biodiversity changes over time. If you sample a particular environment at different time points, you can see how the biodiversity has changed over time. And this can be done, of course, also to assess impacts. If you're building new structures or you're doing mining, you can sample the environment before, during, and after this event, and you can assess if the biodiversity has changed during or because of this impact. It can also be used to finding rare species that you don't easily observe in nature. You take the environmental DNA sample and you can identify the DNA. Having to do biomonitoring traditionally can be very laborious. Obviously, you need to send out biologists into the field and make these observations. So this become a complementary method, you can say, that can be very powerful and save often a lot of time compared to traditional methods. It can also be used to look at diet, for example. I mean, if you sample scats from animals. You can use environmental DNA to see what species that have been prey items for this particular animal, to put that species into the food web and essentially get a better understanding of the ecosystem, for example. That's another applicability. And invasive species is another applicability. I mean, we're very interested in Australia, of course, to see when we have new species that arriving that might be doing a lot of damage to the ecosystem, and this can again be difficult to assess with traditional methods. But if, for example, you take water samples from ports, you might be able to identify new marine species that are arriving as invasive species and might be a hazard to the local ecosystem. That's another powerful, you can say, applied aspect of environmental DNA. And here's a slide that summarizes how big of an impact environmental DNA is now having worldwide. Essentially, you can say you can look at the number of studies that are being published now. There's an exponential increase in the number of environmental DNA-based studies that are being published, with now several hundreds coming out each year. It's across different environments. Terrestrial, marine, and in particular freshwater environments have been studied a lot with environmental DNA studies. This is where it all started with amphibians more than a decade ago. And you can see different taxonomic groups, reptiles, plants, mammals, invertebrates, fish, and so forth. So it's not restricted to a particular taxonomic group. So a very large impact, many studies across the world, and it's clearly recognized now as a very powerful biomonitoring tool with many different applicabilities. And speaking about applications, this slide is simply summarizing 3 examples of publications where eDNA, scientific publications where environmental DNA has really made a difference. Detection of the rare and very cryptic blind cave eel, essentially impossible to monitor. It lives in subterranean groundwater systems, but based on environmental DNA extracted from these water bodies, it can be shown where this species is present. So a very valuable biomonitoring tool to assess the distribution and presence of this cryptic species. Another example in the middle is it was shown based on eDNA of the bird droppings from these black cockatoos that in urban area they 50% of their diet is essentially derived from non-native sources, which is not very healthy for these birds. So many of them might be in a poor health condition because of this. And this has some impact on how to, for example, plan urban landscape design in terms of replanting with native species. And lastly, this has already been touched upon in the earlier slide. You know, you can use eDNA to detect the invasive marine species in ports, where as part of biosecurity surveillance programs. And this takes me to the next slide, which introduces essentially this program, eDNA for Global Environment Studies, EDGES. I think I'll just read this aloud. This is a program that focuses on developing and applying environmental DNA techniques to address challenges related to biodiversity loss and sustainability in our changing world. And that, you can say, that's the scientific objective of this program. But beyond the research, the goal is to translate knowledge into outcomes that will ultimately enhance our collective well-being. And with our well-being, I would also say the well-being of our environment. This is funded by BHP's Social Investment Framework, and it consists of 5 distinct projects that each builds and expands eDNA as a biological monitoring tool. And the overall objective, the outcome should be, first and foremost, this is a research program, so we want to have high impact science and the peer-reviewed papers coming out of this. We also include local and international training of PhD students. We facilitate collaboration and research networks that can expand into larger studies. There's a significant element of community education and awareness implemented in this program. And we also want our research and our results to end up as input to policymakers. As mentioned, the program consists of 5 different sub-projects, covering essentially 4 environments. We have terrestrial, freshwater, marine, subterranean. So really expanding across different environments using this eDNA technology. The 5 projects are a project on the Pilbara olive python for detection and conservation applications. There's a project on subterranean fauna detection and conservation. We have a project on the wetland systems of the Chilean Altiplano. We have a project number 4 that is about improving sensitivity of eDNA tools for invasive species detection. And then we have a project on the benthic communities that are associated with the Griffin pipeline. The first project in the EDGES framework, project number 1, is called eDNA Monitoring and Conservation Genomics of the Pilbara olive python. This project is perhaps a little bit different from the other projects in that it doesn't only include environmental DNA tools, but it also has an evolutionary component, a genomics component included. A few words on the olive python here. The Pilbara olive python is a recognized subspecies of the common olive python. Its olive pythons are very large snakes. They live up to 6.5, or they grow up to 6.5 meters. And the Pilbara olive python has a more, you can see on the map in the lower right corner, has a more southern distribution than the common olive python. The subspecies is endangered, and not a lot is known about its exact distribution and its biology. And this project here is trying to remedy that. In particular, we want to develop eDNA tools that allows us for much better monitoring and detection of this species in its natural environment. Whether that is water samples from rock pools or sediment samples from rock overhangs, for example. Being able to detect the python based on eDNA will allow for much better idea on its distribution, and also provide input to, for example, assessments of local biodiversity, whether these snakes are present or absent. Moreover, we intend to sequence the complete genome of this species, and this allows us to engage in a whole pile of different analysis related to the evolution of this Australian iconic species. How did it adapt to this particular environment? How is it different from the sister species living further north, and so forth? What is the population sizes? How inbred are the populations? There's a whole pile of different questions that can inform conservation management strategies downstream. And with this, I will pass the word on to Nicole White, who will talk about the EDGES Project 2 on the subterranean fauna.
Thank you, Morten. Hello, everyone. I'm Nicole White, a research fellow at the Trace and Environmental DNA Lab at Curtin University. I'm going to introduce to you Project 2, eDNA for Subfauna Detection and Conservation. And this project is in collaboration with Dr. Michelle Guzik at the University of Adelaide and Dr. Nick Murphy at La Trobe University. Okay, for a little bit of background, Australia's subterranean environment is internationally recognized as a global biodiversity hotspot for groundwater fauna. It's also recognized for the large number of short-range endemic invertebrate species and for having one of the highest stygofauna densities in the world. This diversity remains largely undescribed, which is most probably due to the difficulty of surveying or collecting species from these environments. Over the last 2 decades, Australian researchers have and are still discovering a very rich Stygofauna diversity. But despite considerable research in the last decade, much of the fauna remains poorly known. My colleague, Dr. Michelle Guzik, proposed that approximately 90% of an estimated 4,000 plus species remains undescribed in the western half of Australia alone. As I mentioned, given the challenges of sampling these areas, there is a need for the development and validation of meaningful methods that can effectively monitor and assess subterranean biodiversity and these environments. These underground environments are absolutely essential for all life above ground, but as I said, they're poorly described and understood. Given the challenges of sampling these areas, there is a need for the development and validation of meaningful methods that can effectively monitor and assess subterranean biodiversity. In this project, we aim to develop new eDNA tools for subterranean monitoring. How we do that is coupling eDNA sampling with next-generation sequencing, as it has been found to be powerful, time-efficient, and cost-effective. With only trace amounts of DNA required for detection, eDNA monitoring is particularly attractive for the detection of rare taxa. In this project, we will generate a high-resolution mitogenome reference database for selected taxa across geographically sampling points, both from the Pilbara in Western Australia and the Great Artesian Basin in South Australia. This will allow the investigation of intraspecies genetic diversity and differentiation in these taxa in greater detail than attempted before in subterranean environments. The use of this database with the water samples collected from these regions will greatly enhance knowledge about short-range endemism, species boundaries, and evolution in these unique Australian underground ecosystems. In addition, the mitogenomes generated from this project will provide the required molecular baseline for new and optimized eDNA marker development for subterranean biodiversity surveys. And now I'll pass over to Dr. Mattia Sacco to introduce Project 3.
Thank you, Nicole. Project 3 is an international research involving a diverse and multidisciplinary team from 2 prestigious universities in Australia and Chile. By uniquely combining state-of-the-art eDNA technology with isotope analysis, we aim to not only investigate the wetland biodiversity, but also characterize ecosystem web dynamics from sediment microbes to the migratory Chilean flamingos. This will allow us to understand the food chain and identify keystone species that are indicators of wetland health. Specifically, with this study, we aim to survey water and sediment both in space and time for prokaryotic and eukaryotic taxa, conduct hydrological and biogeochemical investigation of the surficial water table, monitor the presence and density of the brine shrimp Artemia, and last but not least, we also aim to survey diets and migration processes of ovifauna that depend on the wetlands for survival. Our expected outcomes involve gaining data on a wide range of species, including the crustacean Artemia, which is a salt-tolerant species of crucial importance for the ecological functioning of the wetlands. We also expect to understand the diet of birds which depend on the wetlands, conduct hydrological and biogeochemical analysis of the lakes, provide new insights on biodiversity and ecosystem structure of the Chilean Precordillera lakes, incorporate a pilot preliminary investigation on the ecology of similar lakes at Rottnest Island here in Western Australia, and ultimately we aim to develop a wetland wealth health index for using long-term environment monitoring worldwide. Now I'll hand over Professor Ewan Harvey, who will introduce EDGE's Project 4 and 5.
Mattia, thank you for your introduction. My name is Ewan Harvey. I'm a marine ecologist at Curtin University, and I've been working on fish ecology and benthic communities for roughly the last 30 years. I have also been working on environmental DNA as a tool for sampling marine communities since about 2014. So I'd like to introduce two projects, Project 4 and Project 5. Project 4 is about improving the sensitivity and specificity of environmental DNA metabarcoding tools for detecting invasive marine species. I'm working with Dr. Justin McDonald from the Department of Primary Industries and Regional Development. Justin is one of the lead scientists for marine biosecurity, in fact biosecurity in general, but as a marine biologist. We also have Tiffany Simpson, who's one of our adjunct research associates, and Shirley Luckhurst, who's our main technical staff member. Project 4 aims to identify how novel environmental DNA techniques can improve marine pest detection in comparison to some of the current methods. One of the things that we actually need to do is to develop phyla-specific metabarcoding assays to be able to better discriminate between what is an invasive marine species and what is a native species. So for example, we could have a range of native barnacles, but we also know that barnacles are one of the challenges that we have with species coming in on vessels. At the moment, we don't have the databases to actually be able to discriminate between them, and that's a real challenge. So we hope to be able to enable the assays to sensitively and confidently detect IMS in the marine environment and to develop the reference sequences for key priority taxa. Those taxa particularly are focused on the Northwest Shelf, and we've identified what species we'll be targeting with help from DPIRD, but also looking at the National Invasive Marine Species Pest List. The outcomes that we hope to get from this project are to sequence genomes from identified pest species and native sister taxa to generate novel assays. We also want to empower eDNA to act as a key invasive marine species biomonitoring tool by confidently assigning identity to pest species by discriminating them from the natives, ensuring that there is accuracy in the techniques when we use them on different substrates. So by substrates, we could collect water samples, we could also collect sediment samples, or we actually might take scrapings from off a hard structure. One of the things we also want to do is decrease the cost of processing samples and also increase the speed at which they are processed. The information that we generate will be publicly available through repositories, and will also ensure that it goes into the global invasive marine species field. By working with DPIRD, we hope that the research will inform policy development about invasive marine species and environmental DNA, and also increase public awareness. Now in Project 5, we will take the assays that we've developed for invasive species, and we will use those for actually looking at the benthic communities that are associated with the Griffin oil field infrastructure with a particular view to invasive species. One of the really neat things about the Griffin field is that it goes across quite a long depth gradient, going from approximately 15 meters through to 200. And this will give us the ability to answer questions about whether there are invasive species associated with hard structures going across the depth gradient, and to actually compare the benthic communities on similar native habitats such as reef. So we'll evaluate the new environmental DNA assays compared to the traditional methods. We'll also validate the invasive species assays where we know we've collected particular organisms. And we're going to document the organisms associated with the subsea infrastructure and surrounding natural habitats. We hope to gain a greater understanding of biodiversity on natural and man-made infrastructure, and refine the technique of sampling biodiversity at depth using environmental DNA. So eDNA for Global Environmental Studies, or EDGES, consists of 5 programs. One is Project 1, which is a terrestrial focused program looking at the Pilbara olive python. We have a second one which is looking at subterranean fauna. This is a particularly important and timely project. We have the third project that Mattia described, which is looking at freshwater systems that are associated with pink flamingos in Chile. I've talked about Project 4, which is about developing assays for invasive marine species so that we can accurately discriminate them in samples from native species which are very similar. And then we want to use these assays for looking at the organisms that are associated with oil and gas subsea infrastructure across a depth gradient, and compare that to the natural communities.